Macropinocytosis is an endocytic pathway that is important for a variety of cellular processes, including cancer metabolism. This protocol allows you to quantify the extent of macropinocytosis in cells in vitro. The automation is aimed at maximizing reproducibility, productivity, and reducing experimental variation. Moreover, the fluorescent microscopy allows you to visually inspect the sample to determine experimental quality and assess additional macropinosome characteristics. Demonstrating the procedure with me will be Cheska Marie Galapate, a research assistant from our laboratory.
For the 24-well plate with cover slip format, use forceps to grip a single cover slip from the ethanol bath. Tap the cover slip to the inside wall of the plate to remove excess ethanol, and place the cover slip flat on the bottom of the well. After the ethanol evaporates, wash the cover slip twice with DPBS. Then, seed the cells on top of the cover slip by adding 500 microliters of the cell suspension to each well. Place the cells in a 37°C cell incubator with 5% carbon dioxide until cell confluency reaches 60 to 80% on the day before macropinosome labeling. The day before macropinosome labeling, replace the media in the wells with 500 microliters of pre-warmed serum-free media, and place the cells in the incubator for 16 to 24 hours. For the 96-well microplate format, begin by transferring the cell suspension to a 25 milliliter reagent reservoir. Then, using a multi-channel pipette, seed 100 microliters of the cell suspension to each well of a black 96-well high-content screening microplate.